Well, thank you very much for inviting me to give this talk. And, one crucial question in the ecology of soil communities has to do with the regulation of decomposers and whether they are mostly influenced by bottom-up forces or rather under top-down control. So many studies have shown how resource availability, both quality and quantity, may regulate the productivity of the detritus community from the lower levels of the food web to the upper ones, so to predators. It's the so-called bottom-up effect or control, but little is known about the role that water availability may play in structuring such communities. So in fact, microenvironmental dampness and soil moisture may control microorganism diversity and biomass, invertebrate abundance and activity, as well as the relative contribution of top-down and bottom-up forces, and then may have the potential for structuring the community as a whole. But animal communities, we know that they are not only structured by abiotic factors or bottom-up forces, but also by biotic factors such as predation. So when consumers or predators regulate the productivity of the detritus community from the upper levels of the food web to the lower ones, we say that the community is under top-down control. In fact, biotic interaction has been shown to be fundamental in the configuration of soil communities, and specifically we think that a predator-prey interaction may contribute to community stability by, for example, reducing the competition among lower trophic levels within the food web. Furthermore, trophic cascades often originate from top predators and can have powerful impacts on ecosystems. So for instance, there are some studies that show how predators can indirectly affect decomposition rates. But although predators can have these large impacts on ecosystem through trophic cascades, not all this effect may propagate down the food web, but there are some compensatory mechanisms that can dampen, minimize, or even suppress trophic cascades. So intragill predation, or IGP, is one of these mechanisms, and occurs when one predator attacks another predator with which it shares the same prey. So intragill predation, or IGP, can obscure top-down control by weakening the potential of generalized predators to control prey densities, and therefore can inhibit trophic cascades. In any case, testing the effects of both bottom-up and top-down forces on the structure and dynamics of the soil community may allow us to better understand the regulatory mechanisms as well as the associated ecosystem processes, and specifically experiment testing the effect of water or the role that water plays in favoring or weakening trophic cascades remains still scarce. So our objectives were to answer these three main questions. So first of all, do changes in the density of large predators affect the abundance and size of the lower trophic levels of the food web? Then, can these potential top-down forces cascade down to decomposition rates? And finally, are these top-down forces influenced by abiotic factors such as rainfall? So accordingly, we conduct a midterm field experiment consisting of the removal or addition of large predators in H beech forest, spread over the Cantabrian mountain range, is in North Spain here. So these 8 beech forests were at similar altitudes, were homogeneous in terms of soil pH, so all of them were acidic, and also homogeneous in the age of the trees in the forest, but mainly differ in the mean annual precipitation. So we have 4 dry forests on the lower mean annual precipitation and 4 wet forest under higher mean annual precipitation. So I think that no, okay. So in spring 2012, and in each of these 8 sites, we established 2 experimental blocks, 100 meters apart, consisting in 5 50 per 50 centimeter plots. And you can see here the blue spots are the wet beech forest, and the orange ones are the dry ones. So we had a total of 80 experimental plots spread over the Cantabrian mountain range. And here you can see what an experimental block looks like. So as I've said, it consists in these 5 plots that are 10 centimeters buried deep in the soil to prevent for horizontal migrations, and had a 1.2 mesh screen on the top and another at the bottom to prevent macrofauna from recolonizing the plots after our manipulations. So 2 of these plots were assigned to the predator addition plot, where the density of large predator was doubled. Another 2 were assigned to the predator removal plots, where all the large predator were removed. And finally, 1 control plot with the natural densities of large predators. So to experimentally manipulate this, the abundance of large predators, and in order to maintain all the animals alive after our manipulation, we did not shift the leaf litter, but rather put it on a tray and look at every leaf to separate and to count and measure all the animals found in each plot. So according to the differences in shape, we separate all the predators which size surpass these size thresholds for manipulation. So spiders larger than 3 millimeters, lithobiomorphs larger than 7 millimeters, and so on. So in the spring 2012, we counted and measured all these animals, and those large predators which size surpass these thresholds were momentarily and individually placed in separate vials until their random reassignment in the experimental treatments. So for each block, 2/5 of the total abundance of large predator were placed in each of the predator addition plot, 1/5 of the abundance of large predator in the control plot, and none in the predator removal plot. Then, to analyze the potential top-down forces or effects of our manipulation on decomposition, we included in each plot in this spring 2012, we included 2 10 per 10 centimeters bags with 1 gram of Corylus avellana of hazel leaf litter each. So we use hazel and not beech because of its faster decomposition for a midterm experiment. So one of these bags was collected after 6 months, so in autumn 2012, and the other in spring 2013, so 1 year later. We know that organic litter decomposes following an exponential decay curve with this equation. So MT is the mass of the litter at a certain time, M0 is the initial mass of litter, K is the decomposition constant, and T is the amount of time passed since the initial measurement. So we can easily calculate the decomposition constant K. So higher K values correspond to faster decomposition rates, while lower values correspond to slower decay. So after 6 months, we reinforce our experimental treatments by a second round of predator manipulations. And in 1 year later, in spring 2013, we counted again and measured all the animals found in the plots. So all the statistical analyses were performed with the statistical software R. Animal abundance, body masses, as well as decomposition rates were analyzed through generalized linear mixed models with rainfall and treatment as fixed factors and site as a random factor. We did not control for block, because when controlling for site, block did not explain any additional proportion of the variance. So we systematically tested 2 models, so one additive and one multiplicative, so including the interaction between the rainfall and the treatment to examine whether the removal or addition of large predator differ among rainfall levels. So to verify if the means significantly differ among treatments, we tested all the models with a multiple comparison of mean or Tukey contrast. So I will now briefly talk about the most relevant results. So first of all, were our manipulations successful? And the answer is yes. 1 year later, in spring 2013, the abundance of large predator was effectively reduced. I have to say that, I don't know if you can see very well, but in all my results, in the left side, we have the control. Here we have the predator removal treatment, and here the predator addition treatment. So as you can see, the abundance of large predator was effectively reduced in these predator removal plots compared to the other two, suggesting that the experimental removal of large predator was successful and still apparent 1 year later. But the abundance of large predator did not differ between the control and the predator addition treatments, suggesting that the overabundance of large predator had been compensated to some extent during the experiment. And now the truly effects of our manipulation. So did this experimental manipulation of large predators affect the abundance and size of the lower levels of the food web? So first of all, the interaction between the rainfall and the treatment was not significant for any of the groups examined here. Here I only show this lack of interaction for the total abundance of fauna, but it was the same for all the groups examined here. And this may indicate that the soil biodiversity or biotic interaction may have attenuated the effect of the rainfall during the experiment. But the rainfall has significant and positive effect on the total abundance of fauna, as found in other studies. So we found more animals in wet beech forests than in dry ones, and the total abundance of fauna differ among treatments. So we found again a reduction in the total abundance of fauna where the large predator were removed compared to the other treatments. So we did all the comparisons among treatments, but from now I will only mention the significant ones. Small or intermediate predators were all those predators which size did not surpass the manipulation thresholds, as well as other groups such as pseudoscorpionids or campodates. So the abundance of these small or intermediate predators also decreased in the predator removal plots compared to the control and predator addition ones, suggesting that the absence of large predator led in the midterm to a reduction of this intermediate predator's abundance. But now let's see what happens with decomposers or with the lower trophic levels of our food web. So surprisingly, we found a negative effect of this predator removal on the abundance of springtails. These animals were 1.6, I think, times more frequent in control and predator addition plots than in predator removal ones. And in these plots without large predators, we found that the size, the springtails were larger. So we think that to some extent, the largest species of colembolans are being favored by the absence of large predators. Similarly, the abundance of oribatid mites was also lower in the predator removal plot compared to the other two. And on the other hand, julids, these strict decomposers, were more abundant in the plots where large predator were added. So suggesting that this addition of large predator favored the presence of these strict decomposers of julids. And now the ecosystem effect. So did our experimental manipulation of large predator cascade down to decomposition rates? And the answer is yes, because as you can see on the y-axis, K values were lower in the predator removal plot compared to the other two, suggesting that the absence of large predator, far from enhancing decomposition, have led to a slower litter decay. So as to discuss a little bit our results, our manipulation were successful and still apparent in 2013, at least for the removal of large predators, and we found a surprising negative effect of this predator removal on the lower trophic levels within the food web. We think that under lower predation risk by these large predators, intermediate predators may have initially increased in abundance, which may have led to a decrease in prey abundance. So then in the midterm, This may have led to hunger and even starvation within these intermediate predators, and may have decreased in abundance in this time. So this points out the positive role of large predators in regulating the lower trophic levels of the food web. And this, the negative effect of the predator removal on litter decomposition rates can be therefore explained by this reduction of decomposers in the plots without large predators, by this higher abundance of strict decomposers in the predators where large predators were added, and this higher abundance of the larger species of colembolans that can maybe belong to the family of tomotherids. We know that tomotherids can overgraze on fungi and can retard litter decomposition rates, so this can maybe explain these patterns. So as a summary, we think that a large predator may exert a very strong control on intermediate predators, therefore indirectly and positively affecting decomposers and as well as decomposition rates. We want now to test this direct versus indirect effect on this of this stress to uncover what kind of mechanisms can underlie these results. So, as to conclude, the absence of large predators led to a decline of prey populations, a decline of intermediate predators, and slower decomposition rates. So I want to thank all the co-authors of this study, as well as all the people that helped in the field, and thank you, all of you, for listening.
Thank you very much. Questions?
Thank you for the presentation. Like, you have big, like, is that variance or error?
Is the confidence interval.
Yeah, there are very people. I think the experiment has a lot of like ecological realism, well, in my opinion. But I was wondering whether you think like the absence of predators can increase actually competition between the other taxa, and that's the reason you have lower abundances.
They can, this competition because of the absence of large predator. I think that when this absence of large predator may lead to initial increase abundance of some intermediate predators, and maybe yes, competition. But I think that these results points out that this large predator really positively regulate the lower trophic levels and the whole food web. So.
Yeah.
I don't know. But.
There was this. Yeah. Thank you.
Okay. Oh, we have another question at the back, at the top, please.
Thank you for the talk. Just about the interpretation of your result. Maybe you don't know the paper of Michel Laureau and Claire de Mazancourt, which illustrates the fact that with herbivore, it's not the same, it's not a top predator, but it could be the same mechanism in my mind. There is recycling loops which are stronger or more numerous, and so it could accelerate the decomposition of organic matter. It could be an explanation.
Okay, yes. Thank you very much. I will take a look of this study.
Thank you.
Thank you very much.
We have one more question.
Thank you very much. I think I'm going to make the obvious question, and sorry if this was clear and I didn't get it. How did you control for manipulation?
Control for manipulation? For the I had control plots where the density of large predator were not manipulated. So I have 5 plots, and 2 were assigned to the predator addition plots, 2 others to the predator removal, and then in each block I have 1 plot where the density of large predator was not manipulated.
And the box?
The box?
Yes, because the manipulation of the predators involve also the isolation box, right?
They are not actually isolated, because it's a 1.2 mesh screen, so it allows most of the animals to trespass this netting. But you mean that in the control plot I haven't manipulated in the same way that in the others? Yes, we have not separate the large predator, but yes, manipulate the leaf litter to have the same effect of our manipulation on the plots. So
okay,
we did.
And you had those as well with the boxes, even if they were not?
You mean a control out of these boxes? No, I don't have this control. But we, as I said, we did the same. We take all the leaf litter with us and counted and measured all the animals in the same way than the others. But yes, we don't have this external control.
Just to gather how close to the natural situation you are in
It's
your
a very,
control.
this experiment is now going on for a 2nd year, and now we have the results of our, and we, if you see, we don't have a decrease in the animal abundance 1 year later. So I think that what we found at the beginning was not significantly different in terms of abundance of fauna 1 year later. So this can suggest that our experiment was natural and was not damaging the food web. Or
especially I was thinking about this because of the springtail reaction. They are obviously very sensitive to anything you do to the litter, particularly, you know, if it has to do with drying it out, and or limiting the movement of water.
Yes, but if we have manipulated all the plots in the same way, we should have found that these springtails were also lower in the other treatments, but it's not the case.
Okay, and my other question was, I was wondering, because you said you were using multiple comparisons, post hoc multiple comparisons, and you did lots of them, and I was wondering whether you used any method to control for that?
I don't know if I am following you.
Well, you did many multiple comparisons.
Yes, among all the treatments, so control
yes, and for many variables. And I was wondering whether you used something that to control for the inflation of your.
I haven't put the result of the total model, but I also test not the comparison among treatments, but also if the abundance of groups or the size of groups was significantly different among rainfall levels and treatments.
Yeah.